Welcome to Analytically Delicious, Insights into Food Safety by LVA. Join us as we explore the latest developments, trends, and challenges in ensuring the safety of our food supply through analytical techniques, uncovering the science behind what makes our food safe and delicious.
Welcome everyone to another episode of Analytically Delicious. I'm your host, David Steiner, and in today's episode we dive into the critical topic of allergens in food safety. Allergens are not just labels on packaging, they can pose significant health risks for millions of individuals worldwide. From gluten to peanuts, allergens are a challenge for food producers and regulators alike. But thanks to advances in analytical science, we can better identify and control these risks. Joining us are two leading experts from BioCheck UK, Adrian Rogers, Development Manager and a specialist in immunoassay development, and Richard Fielder, Business Manager with extensive industry experience. Together, they bring decades of expertise in allergen detection and food safety solutions. We will explore the science behind allergen detection, the tools and techniques used in industry, and we will look at why harmonization in testing methods remains a challenge. Whether you are an industry professional or a curious listener, this episode promises an engaging mix of scientific insights and practical application. So Adrian, Richard, welcome to our podcast. It's really nice to have you here today.
Thank you, David.
Yeah, you're welcome. Thank you. Yeah, thanks for inviting us.
So before we dive into today's topic, which is all about allergens, allergen testing, let me highlight one thing. Actually, a funny story we all share is that we all have a history with Roma Labs, and obviously, aside our passion of keeping food free from harmful substances and also talking about the analytical stuff behind that, it seems that Roma Labs has some kind of cornerstone in all of our careers. And now I'm really curious, Richard, you are now with BioCheck for more than 1 decade, 11 years. So what does the company specialized in, and what are your main responsibilities there?
Thank you, David, for that introduction. It reminds you that it's a small world, isn't it? And I think a lot of us do move between these different diagnostic companies. I think I've been working with Adrian for 25 years. BioCheck is indeed a specialist company, so a little bit different from the other test kit manufacturers. So we're just focusing on food allergens and meat speciation and mycotoxins as food safety topics. In that, we're developing and manufacturing antibody-based techniques for food allergens, and my job has really been for the last 3 years is to run the company and to try and understand what's happening in the market, some of those trends which I think we're going to talk about today, what we've been trying to do is put more investment into the food allergen side, because there's an awful lot happening and emerging trends coming along as well, which we're looking at, we need to address in the future. One of the key things we're trying to do is look to ways of helping people. It is a specialist area, analysis, and it's just trying to use our knowledge in this area to try and help people, tackle the challenges they see.
Mm-hmm. That's a very nice motivation, actually, to stand up every morning and go to work. Yeah, and
it helps.
Adrian, can you maybe share how your role as a development manager at BioCheck aligns with the mission of the company?
Yeah, sure. So I've been with BioCheck now for about 3.5 years, and as you said, it was at Roma Labs prior to that, and another company, whereas with Richard prior to that as well. So we've got a long background and experience with developing immunoassays for allergen detection. So from my side, I think the main thing is, I develop the methods, I help develop the methods. So I'm the perfect person to tell a customer how to get the best out of the methods. So it's all, from my point of view, it's sort of developing the methods, but also helping to support the customers a lot. And there is an aspect of hand-holding to some extent, and I think you need that. This, as you'll hear as we talk through, the things that arise and the issues with allergen analysis is very complex. It is a complex procedure and process. So I think people do need, their hands held a little bit, and how to get the best out of a method. In our case, an ELISA kit or a lateral flow device. I don't believe in the fact that they just have to. You give the guys a box and then run off as quick as you can in the opposite direction. It doesn't really work like that anymore, and I'd feel it wouldn't. We'd be missing a duty of care, I think, if we did that to our customers and just say, no, here you go, here's a method, you get on with it, and we'll see how you get on. It's a lot more complex than that. So if you can help a customer sort of firstly understand how the method works, but also the method might not suit their needs, so there might be a bit of tailoring or a bit of customization that would need then help them get the best out of that method and get the either more reliable results or results that are better suited to the different foods they're trying to test.
Mm-hmm, mm-hmm. So it's basically also a lot of consulting work and of course also support of the customer to establish a method in their own facilities and so on. So it's not just to establish a test kit and sell it and say goodbye, that's it. So it's really also customer care. Yeah.
Yeah, I think that's a potential. Well, it is a theme we're like to get across today, is having that dialogue with whether it's your lab or your kit supplier about what you're trying to do, rather than just buying the product, because I think there's a lot more support they can offer you. If you take that step of, it works two ways. It's equally for manufacturers and labs to reach out, and also for the customers. And I think that really does help then.
Absolutely, yeah. So Adrian, you already mentioned that allergen, the whole topic as such, is very complex and a very broad topic. Maybe we start with I would say, rather basic question. Maybe can someone of you start to explain what makes a food component actually an allergen? And what is the mode of action behind an allergic reaction?
Yeah, perhaps I'll just start there, because I think you introduced the word complexity and also the word broad topic, and I think that's it. There are a whole range of adverse reactions to food, of which food allergy is only one of them. We talk, in fact, about food hypersensitivity, because we also recognise people have intolerances. Some of the immune mediated reactions are actually not IgE, which is the underlying nature to a food allergy, but actually autoimmune. So if we talk about gluten and celiac disease. So in fact, we are talking about a whole range of different adverse reactions to food. But I'll probably start in a simple way, and I'm sure Adrian can add the complexity.
Well, we'll see. I'll add, we're not immunologists by trade, so you'll have to excuse us in some of our definitions.
Yeah, probably to answer your question, we do actually need a team of people, and many decades have been spent trying to understand, this very question. But I think what we could say is, and probably hopefully everyone would agree, is that in terms of when we're talking about food allergy, a food is allergenic when it contains a protein to which a person's immune system reacts. And I think that's, at a basic level, we should all be able to deal with, be comfortable with that. I think the other side of the coin, as we would say, or the other side to that, is not all food proteins are allergenic. So actually, and then it's trying to, well, why not? And what makes, so your question is a really poignant one.
I'd question that. Any food protein can be an allergen. No, it's true. Anything can be an allergen if it's a protein, but it just depends on the prevalence and some proteins are more allergenic than others, and that's maybe where we don't quite have that full understanding. And this is where there's a lot of research going on. So, for example, with pea. Why is a pea not quite as allergenic as a peanut? They belong to the same family, and I know that that there's more conversation to be had there, because pea is one of those things, and we'll talk about this later probably, sort of on people's watch list as a growing allergen. But it's because it's happening now because of the sort of meat substitutes and more vegan and vegetarian diets. So they're looking for substitutes and they're looking at protein substitutes. So they're using pea protein a lot more in food, and you'll see this as a trend. Sorry, I'm stealing Richard's thunder. You'll see this more of a trend as different foods get introduced. As the diets change, then you will get a subsection of the society that will start becoming more allergic to that.
No, Adrian, I'll feed you more lines to react on.
No, you carry on. Sorry.
I think one of the points, just to pick up on what Adrian was saying there, is the amount of the allergen is also critically important here in terms of it's the dose. At a certain level, we get a reaction, and not all people are the same, so that dose will vary. I think also the structure of the proteins, we know, heating, processing, it alters the structure, so that can make them less allergenic, more allergenic. And those are other aspects with, there's a lot of research trying to understand that. And, we know that these proteins, actually, the ones which are allergenic are quite robust, some more robust than others. If we look at peanut, AROH2 versus, some of the other peanut proteins, AROH6, 9, whatever, then, there is difference in thermal stability. And that's really important when we talk about detection, because we need a marker which is going to be thermally robust to the process, so that and something which we can recover, solubilize, etc. But the other aspects in here, just to say, but there's a lot more cofactors. I think I know Adrian probably want to add in on this point, which makes food components allergenic.
Well, yeah, I would say it's not a simple way of saying, if you have so much peanut, then you'll have a reaction, because it very much depends on the individual. And like Richard said, these cofactors, like you can get exercise-induced anaphylaxis, so you're more prone to have a reaction after you've exercised. The levels of hormone, if you've been out for a beer the night before or whatever, and all these different things can interact and have an effect on whether you have a reaction or not. So this is everyone's individual, and this is what makes setting a threshold level so difficult, because everyone reacts at different levels. So you have to take it from a population view. So you say, 95% of the allergic population will be safe at such and such a level. So that's the way to do it, and it's very difficult then to tailor it to an individual's dose. You don't know how many peanuts they can eat and be safe. So that's sort of the tight line we're treading with this a lot of the time.
Mm-hmm, mm-hmm. And how would you say has scientific understanding of allergenic proteins evolved in recent years?
Well, probably in every conceivable way, really, because I think the whole area of that studying of proteins, the proteomics, we've learnt an awful lot about the structure of them, their function, and I think for the people working in this sphere, that's helped them in their work, whether you're an allergist in clinic or an immunologist looking at the antibody side, whether you're a biochemist really trying to work out, what's the antibody doing, will it react, how does it react, is that binding, is it, what's the affinity of it, but at the end of the day, all this is to help the risk assessor ultimately say, right, we're going to label this product in such a way so we can communicate, so the consumer can be better informed. So, there's been advances, I would say, in a lot of different areas here. There's a lot of studies which have gone on, and perhaps Adrian might say something a little about some of those studies to advance this knowledge.
Well, I'll just say a bit about if you know about the structure of a protein, then there has been trials run and work done to try and make them less allergenic. So if you know what causes the allergy, then you can sort of breed that out and make a less allergenic nut. So I know there are trials, etc., going on with that, but also how this doubles back into the analytical side as well. If we have knowledge of how a protein binds to the antibody in a person, then we use antibodies in our methods, they're immunoassay methods. So if you know how they bind specifically to the proteins in a person, then there'll be a similar sort of, I'm not going to say exactly the same, but a similar sort of mode of action that happens on your antibody plate. So that's why you can see sometimes similarities in cross-reactivity. So if you've got a walnut antibody, you might cross-react to pecan because they're from the same family group. But there may be an instance where someone with walnut allergy also has a reaction to pecan as well. So there is a sort of, like I say, I emphasise the fact it's not exactly the same, but there is a sort of mode of thinking there that there's similarities. And just going back to the allergy side of it, it's very regional. I think what people don't quite understand, it does, depending what part of the world you live in, you can have different prevalence of allergies. I mean, we looked at that a few years ago. I was involved with the Your Proval project that looked at that, exactly that, the prevalence of food allergy across Europe. And they did find where they thought, it'd be the same. Everyone eats the same food. It will, prevalence would be the same. It wasn't. There was a lot of localization. And that knowledge has then led to possible, you can never cure allergy, but possible treatments as well. There was a few years ago, there was something called the LEAP study, which was learning early about peanut. And this made quite a lot of news. And it all came about because they noticed that children in Israel, or population in Israel, didn't tend to have a lot of peanut allergy. So they couldn't quite understand it. So when they went to Israel and they looked around and looked what people were doing, there's a snack you can get in Israel called bamba, which is a corn snack, puffed up corn snack that's coated with peanut, and a lot of people eat it, and it's a popular weaning food in children. So, as soon as they start crying or start teething, they start putting the bamba snack in their mouth, so they get this early exposure to peanut. And this was a cue then to say, well, should we be feeding, potential allergenic foods earlier to children? So when they start weaning, because there has been a thing since the 80s, let's say 80s, saying, you shouldn't give allergenic foods early in life, and especially with nuts, because they're classed as a choking hazard. So you can understand why. But there was a big, and especially I was just thinking back to my children are now 11 and 9. When they were born, we were instructed, to withhold weaning until a certain time. So there has been a change now with weaning guidelines to introduce more foods earlier, and this is a way to build up more protection from allergies as they see it. So that's it. So this is a lot of understanding about how the nature of allergy and how the early exposure can lead on to development of allergies. But trust me, it's not as simple as that. There are lots of other cofactors involved as well, but it was quite an interesting study and a story behind how it came to be as well.
Yeah, so I think we're talking about, regional differences. We're talking there about age being important. So children, we'd see different prevalence of like milk and egg allergy. My youngest son, he had an egg allergy, but he's grown out of it. We see that actually the history in the family of allergy is important. And, we see that allergy in certain populations is higher in some countries than in others, like celiac disease in the Irish population compared to other countries. So I think it just illustrating the complexity here. And that exposure is also, whether that's, we're talking about ingestion, but also through the skin, so there's a lot of factors and considerations.
Mm-hmm. Yeah, definitely. And thank you very much for these very broad insights also. I would like to shift gears now a little bit and go into the direction of the analytical part behind this topic. So we know that in the area of allergen testing, especially immunological based assays are most prevalent, I would say, very dominant in the market, like ELISA or lateral flow devices. Could you briefly explain the differences and why different approaches are actually needed?
Yeah, I think if we start at the sort of the broadest level, what is determining what we're trying to do. In some purposes we're trying to screen, and other purposes we're trying to confirm, and that might be because you're doing an investigation, or you've got a result and you want to use another technique in which to confirm whether you're getting the same conclusion. So having that range of different methods, of which the immunological, and particularly ELISA, that's a gold standard method really in food allergen analysis. But the other techniques, the PCR, DNA-based techniques, even NGS these days, next generation sequencing, play a role. And increasingly, as all of us know, mass spectrometry has grown really in the last decade, amazing powers, but more as a confirmatory tool rather than on a screening. And certainly also lateral flows have been transformational in their use.
And what are the biggest limitations to these analytical techniques?
I think there's quite a few. I mean, it's going back to the sort of previous question a bit. It's good to have a toolbox. We always advocate having a toolbox, whether that's you've got the different methods, ELISA, lab-based methods, ELISA, DNA, mass spec. But I mean, with all those, you still got to get the particle of interest out of the food in the first place. So that is a huge undertaking, to be honest. If you think about mass spec, you can't just take your food, stick it in the mass spec, and hope and cross your fingers and hope. If that happens, you've ruined €500,000 of equipment, because they're not designed. Yeah, they're not designed to do that. So there are steps to be taken to process your sample before you can put it into the method of analysis. And the same is true with ELISA. Even the lateral flow devices, you've got to try and get the protein out of the food in the first place. And that is where I see what could be the biggest drive to improve allergen analytics. And it's something we're looking at, is the best way to get the food out. Because, sometimes you think, you want quite harsh reducing conditions or whatever, but then you've got to think that would then affect your assay, because you're putting the antibodies would also be affected on the plate, or the lateral flow device would also be affected by the harsh conditions of extraction. So there's considerations. I always see immunoassay as a balancing act. You can never quite do the best thing to get the protein out, but also do the best thing to get the best way to detect the protein, if you like. There's a bit of a duality. You just want to get the protein out. The harsh conditions may be needed to do that, but those harsh conditions aren't then compatible with your method of analysis. So it's about a bit of a compromise and a bit of a balancing act, and it's important to know about these things. I mean, the classic example with ELISA and lateral flow devices is chocolate. It's very difficult to get proteins of interest out of chocolate. You can look at, if you're doing spike recovery experiments, say with milk, you can put a known amount of milk in, and you can guarantee you can only get about 50% of that back out. And it's because that the chocolate contains polyphenols, these tannins, and they hold on to the protein. They don't like letting go of it. So one way around that is actually adding in more protein, so the tannins stick to that and hopefully release your protein of interest. In this case, the milk proteins, release them more freer into the solution. So that's sort of an area I think needs to be looked at a lot more. Is the extraction part? It's getting those proteins out. If you think about lateral flow devices, ELISAs, they've been around 30 odd years. There's not too many advancements in the actual technology of detection. Yes, you can change the platform, as in put the antibodies on something else. Surface plasma response had its day a couple of years ago. There's lots of ways to apply the antibodies, but the whole thing about an ELISA, it hasn't changed that much. What you need to look at there is how to get the protein out, and maybe raise your antibodies against more specific targets. I mean, back in the day, people just wanted a test for peanuts, say. So they'd go and they just mush some peanuts up, stick it in an animal, raise your antibodies, and you got it back. But that's great, and that worked for a while. But then you've got things like soya. Well, if you think about what form of soya people eat. People don't eat raw soya. So your method you developed 20 odd years ago against a soybean extracts, a pure, then it's not as relevant for its use, its commercial use. People eat a processed form of soya, and even more complex than that, soya fermentation products. And if you think about soya sauce, it's soya sauce. It contains soya. If you had it in a food ingredient, you've got a label for it. You can't detect it with a soya assay because it's that highly processed. The proteins are chopped up that much through the fermentation process, so your antibodies can't actually detect it. So there's things like that. There's these nuances and things like that to sort of take into account that make life very interesting. And I did have nice dark hair when I started this, and it's going a certain shade of grey now over the years I've been dealing with food allergens. But this is how, talking to kit producers, talking to experts does help. Don't think as a user of analytical methods or whatever, or if you in the service lab, reach out to people, understand, because we've been doing this a long time, and there are these idiosyncrasies that happen with allergen analysis, and it's important to learn and propagate that information.
So maybe to conclude at this point, the analytical challenges are not here now based on the detection part. It's more the extraction, what you said, and it's also about choosing the right matrix, which then reflects the real life condition, right?
Yeah, and I think at the end of the day, it is trying to verify whether the method will work with your samples. That's so key. Purchasing a test kit, yes, a test kit manufacturer will provide data, but actually what's more relevant to you is your samples and how that test kit will perform. I mean, the other thing I would say in here, which, Adrian's talking about, the need to try and tailor those methods. And I think, ultimately that comes down to trying to get reliable results. And that's what we're all interested in. At the end of the day, what is the true result? And I think, we talk about rapid methods when we talk about lateral flows. And I think for me, that sort of misses the point. Yes, we do need to do it quickly, but there's no point in doing it quickly if you're not getting a reliable result. And sometimes, and it's so important because, the whole on-site testing side is expanding so quickly, and it's getting people to actually remember sometimes a little bit more time on the very points Adrian's been saying. The extraction side will lead so much more to a more reliable result in terms of the detection. So
I was just about to interject there. It's not just the extraction side; it's the sampling to start with.
Yeah, well,
so this is one of Richard's little bugbears, which I'll let him go on a bit more about this. And it's the side that's not looked into that much. It's not researched that much.
No, I think let's face it. All three of us, we've worked with mycotoxins, and there's a lot more work that's been done over the decades on sampling, and there are sampling plans. Whereas we don't see that, with food allergens, and we have. They're similar problems yet different. With mycotoxins, we get hot spots, but obviously a mold can grow in a silo where there's humidity and you get the toxins produced. The allergen, it's either there, it doesn't multiply because of a fungus. It's either present or it's not. So it is about trying to detect those hot spots in a batch. So sampling is so important. And we don't really have the numbers on the variation around sampling, but we know in mycotoxins, 80 to 90% of that variation, analytical variation, can be attributed to those early stages of sampling and sample preparation. I think when you look at mass spec, I think we can differentiate the mass of an ion to 4 decimal places. That's the power of detection. But we've got to get the sample right.
It doesn't help you to be that precise, if you have the wrong sample in house,
If you think about the uncertainty of an ELISA kit, if you look into mode of uncertainty, sometimes it can be plus or minus 50%. So then recording your result to 3 decimal places is just nonsense. But people do it, and this is something that. Well, I know we're going to look about harmonization, but it's key. It's key that everyone sings from the same hymn sheet, if you like, in how to report results. and so that they make sense, not just to the person producing the reports, but also how they're interpreted by the customer with those reports as well.
Yeah, Adrian, you already mentioned the term harmonization. So I think a recurring challenge in allergen detection is the lack of harmonization. And for me now it's interesting, why do different countries actually or labs rely on different standards and methods?
Well, I say it's not as simple as that, because you can be a producer, you can send your sample to 2 different labs and you'll get 2 different answers. And that's the upshot of it, and that's what causes a lot of confusion, and especially in the case of if you've got something like gluten, where there is this 20 ppm level. What happens? The one lab says it's 15 mg per kg gluten, but the other lab says it's 25 mg per kg gluten. Who do you believe, and what how do you proceed from that? So if you go with one lab results, yes, it's okay, I can sell this. If you go with the other lab result, no, I've got to do a recall because it's over a limit. So this is where there's a lot of confusion there, and this is why we talked about some degree of harmonization. I mean, it happens. I mean, to understand why it happens, different kit produce, because there's no. A lot of it boils down to reference materials. With mycotoxins, you have this set of reference materials. Well, those aren't there. Well, so much with allergen analysis. They're getting better. For 20 years, we've talked, we need reference materials, but no one's done it because they want it to be perfect. Well, there's no such thing as a perfect reference material for allergens at the end of the day. And this is what put people off. So now we're of the thing, well, we've got to do something. So we've got to put something else. Having something is better than nothing. And this is sort of where we've started from now, is having a material that's well characterized and is used by people. And that's the biggest challenge, is if the reference material isn't used by people, then it's not really a reference material, because people don't use it to reference against.
Well, I think the biggest issue with the reference materials is that you don't have a proper reference method as such, right? I mean, there is nothing like a mass spec method established on the market, and that makes it quite difficult to establish this reference point, right? I think there are a few reference material on the market, but not very successfully established, I think.
Well, commercially, yes, I think there needs to be a lot more uptake. I mean, LGC and the Food Standards Agency worked on a true reference material produced to ISO standards, 17034, I think it is now. So there are standards there actually helping to get that consistency. So, to be a certified reference material, we need that traceability back. So that's another level of quality involved with that. But we do have a material now which is milk, egg, and peanut. Was it almond and hazel and walnut? I think it was. It was 5 allergens, but I'm sure LGC will take your calls to provide more details. And Monica did some work as well. So for me, I mean, I think there's a role for standardization on the path to harmonization, if that's not too much of a sentence. And there are global standards. If we think about Codex and Codex's role, we've had a global standard for pre-packed food labelling for over 40 years, with listing the priority allergens. We have labs working to ISO 17025. So the approaches are getting standardized, but I think there's elements of analysis which would be really, really important to standardize, not least the units of measurement, the reporting. We've got to say it, which is, I think the consensus view, milligrams of allergenic protein per kilo. Or provide the conversion factor. So if you're doing peanut, reporting is milligrams of peanut per kilo, give a conversion factor so you can convert that to peanut protein. Because I think what we see problems in proficiency where they have to report the numbers given by the labs, and are we mixing up sometimes the units of measurement? I'm not saying that's the case, but there's the potential there always, and certainly when we're communicating. And I think going forward, if we don't get the units of measurement the same, when we use tools in the future, such as AI and it's scavenging the internet for data, we're not comparing like with like. So, that it is really important, particularly if we're going to have thresholds for precautionary allergen labelling, which is really the sort of the driving force at the moment for the harmonization, because we have mandatory labelling of allergens for ingredients, but it's the whole may contain, that unintended presence of allergens where, it's voluntary, it's inconsistent, and that's not helping the consumer who's got a food hypersensitivity. So, and that's where analysis can really play its role. If we have levels and we all start using some standard
I think that's it. It needs the take up. It needs people to be invested into the whole process, and you can't work in a little silo by yourself and cross your fingers, hope for the best. You need to be part of a bigger discussion and getting. To that end, we try and position as BioCheck, Richard and I try and position ourselves on as many boards and working groups as possible, just to try and get the dissemination of information. To that end, there's a lot of different groups working together. So AOAC have just republished their Appendix M, so looking at how to validate ELISA kits. We're currently trying to work on end-user guidance, so with the AOAC as well. Richard, I know you made a list earlier of all the different working groups that are trying to work towards harmonisation. But what I will say, it's great that everyone's trying to harmonise, but your risk assessor in your small to medium enterprise doesn't want 5 different folders, 5 different booklets saying, oh, this is how to do it. This is how, we're harmonizing it, but then everyone else has produced a different report saying how to do it. So this is something we've raised as well. Sort of, if we go, don't reinvent the wheel. If there's other groups doing it, then work with those other working groups to share that information. So, Richard, I don't know if you want to get a quick word about the other different groups.
We see different groups, particularly in Europe and in America. So AOAC, we've mentioned, doing a lot of work now, very active, and actually that's pulling people from across the globe. Because Australia, New Zealand have had done tremendous work. That's through the Allergen Bureau with the vital standards. They're involved in discussions, as well as obviously the regulatory authorities, FDA, or in Europe we've got EFSA and the Joint Research Centres collaborating. At CEN, which is the sort of European committees for standardization. Working Group 12 is for food allergens. We've come out with a number of standards to support best practice around immunoassays, also with minimum performance criteria. What makes a good method. Mycotoxins, that's an approach which was taken and has been very successful because that encourages new methods to come along. In the UK, we've seen a long history of different groups, but more recently we've got the MFAN meetings, haven't we, Adrian? Which is through Professor Claire Mills. Perhaps you want to say something on that group.
Well, it's a way of bringing people together. So it's a way of getting all stakeholders involved. So be it analytical labs. Legislative bodies, so the FSA in this country, kit producers, and the retailers, and even the allergic consumer. So it's a way to get people talking together about what the issues are. And this is again the only way. I see it, and I'm sure Richard sees as well. The only way to progress forward is to have the conversation with everyone, with all stakeholders, and try and move things forward in that way.
Yeah, linking up actually that consumer side all the way through, manufacturing to clinicians, with the analysts and risk assessors. That's really important. I know in the UK we've got, for example, the Anaphylaxis UK. They have a business forum, and that's part of what they're doing, getting that dialogue, the voice of the hypersensitive consumer and feeding that back into industry. That's really good to see. there's other groups on gluten. There's the prolamine working group. In Europe, we've got a network of food allergen laboratories. They're trying to harmonise. They had a meeting in September in Italy, and they're trying to standardise their approach to work. So there's an awful lot of work going on. I mean, in the Netherlands, they've just introduced a new ruling for may contain labelling, and there's some guidelines around methodology to support that, which was released as well by the University of Wageningen. So there's a lot of information. I think what this sort of discussion highlights is, how do we get it out there? How do we, all of us, help disseminating it?
We have now already talked quite a lot about analytics already, and I would say also about basic allergen testing. I would now like to talk a little bit about the health implication, what the analytical science has basically. So how does analytical science help to mitigate risks for consumers with allergies?
Yeah, I think, perhaps two points we could say here. I mean, we have free from foods, and clearly analysis is instrumental in helping to deliver not a product which is free from in the true zero tolerance sense, because in analysis, we can't measure zero, but we can report less than. And I think, with gluten, it's well established, less than 20 milligrams per kilo is the regulatory threshold in a lot of countries and at Codex, so we can work to that. I mean, I think the other aspect is, which is what we've just been talking about, is the precautionary allergen labelling, the PAL labelling. That's the whole driving force at the moment, and how we can get more consistency by setting thresholds. If we have a number, then we can start to try and work to it, as long as we understand what that number means. At the moment, we have a debate around threshold doses, because a dose then needs to be interpreted in terms of analysis to a concentration. In other words, what is the dose in a serving size? So, it will be, in a teaspoon versus a ready meal, then the levels clearly will be different, higher in a teaspoon than a ready meal to deliver the same dose to have that same effect. So that's part of the discussion at the moment around EDO5s, EDO1s. So that's at the population level, how do we protect 5% of the population or 1% from the most severe reactions by implementing such a system?
Now, what I will add, though, is the most sensitive test isn't always the best one. I'll just point that one out, because there is a bit of a drive sometimes. So, this kit can detect 1 mg per kg. But this kit's better because it detects 0.5 mg per kg. It doesn't always correlate like that. And it's about, if you think about the way an analyzer works with, you're measuring an unknown against a set of known standards. But those, you hope those standards form a straight line. But if, and as you know, that the closer to zero, then the more uncertain you are of the result. So it's best to have something further up the scale to get more consistency with the number. And like I said, once you start thinking about measurements of uncertainty, et cetera, it does. You start realizing, you put a number on a report. I know we're going back to analytical again. I'm sorry, but it's just one of those things. It's all well and good putting a number on a report, but it's knowing what that number means is half the battle, to be honest. It's interpreting and making sure that customer, the food company that's commissioned the testing, knows exactly what that number means to them.
Yeah. So I think it is back to this point about it's clinically relevant levels. That's what we should be. And that's the move now. That's why we talk about what are the doses at the population level to protect. And in countries such as the Czech Republic, since 2018, they've had levels for PAL labelling based on analytical capability. And over the years, people have chased sensitivity, and I think that's certainly a move Adrian and I would like to see addressed, to bring it back to actually what's clinically relevant and making sure we can get robust, reliable analysis at those levels. That would be, really should be a key drive going forward, and I think is the direction of travel now.
And do you have any specific examples where improved detection methods have led to better outcomes for consumers?
Yeah, I think one of the biggest changes in more recent years in testing has been with those lateral flows. It has literally, I think, in analysis, we've seen 3 paradigm shifts. We talked about mass spectrometry. That's a bit like doing a protein fingerprint. The chemists will probably hate me for saying that, but that's the power of mass spectrometry has been transformational. The same with next generation sequencing. And the other one is lateral flows, that whole on-site testing capability. Being able to do really what a lot of factories like to do is do that screening on the factory floor. So when they're trying to say, is this production line clean and I'm ready to set up for this free from product, or just a product which doesn't contain allergens which were in the previous product, giving that assurance so that they don't progress to the next step until they're sure that everything is right. And that whole philosophy, which is that sort of HACCP base, that's where the lateral flows really do have an important contribution, and I think their potential is going to grow more and more. There's, as a platform, there's so many different ways we can use them, particularly with new technology and cloud-based technology as well.
Yeah, I think that's it. You see, looking to the future a bit, and sort of, AI is the buzzword at the moment. But Rich and I have long discussions about this and how you can implement it. But it's if you gather enough information about hotspots in your factory, or if you're a multinational, hotspots of allergen issues across the number of factories, you could end up in the position where if you feed all that information in, to be able to start to predict where on your production lines you might actually get issues, and actually preempt that those certain areas may need more cleaning or more regular clean downs, and so that helps then ultimately cut down out the number of product recalls that you get, and also improve your efficiency, because you've sort of negating out where these possible issues might happen, and these allergen instances might occur within your factory.
Yeah, that's really exciting potential when you start thinking about it. Being able to direct testing where it's really needed in a factory, because you can really see and predicting those trends. And as we see in the supply chain, all sorts of trends. If you can predict in advance problems as they occur, and linking these days importantly to other factors, whether it be changes in the climate, drought, shortages, infestations with different crops, which could lead to shortages, so that another product might be used. These are all problems we see, and we have seen only over this last year. That would be hugely powerful, and just turn really the power of analysis on its head to be more of a sort of strategic tool in its use.
So since we are focusing on food safety, let's look at the regulatory side. In Europe, we have strict regulations on allergen labeling, right? Do you think these regulations are sufficient, or do we need to take further steps in terms of allergen risk management? What do you think?
I think what we've seen is a number of calls for what can we do more in terms of control, whether it be at regulatory level or through industry-led initiatives. And certainly we're back to precautionary allergen labelling again, because that really is the topic. And it's seeing how, in the absence of, at the moment it's voluntary and it's inconsistent, and that is confusing for the customer, ultimately for the consumer. And in some countries, in Europe, we've got, although it's meant to be a common legislative framework, we spoke already about the Czech Republic since 2018. Germany doesn't have it at the regulatory level, but they do use action levels based on clinical thresholds to help inform them with analysis. But on the regulatory side, we see in the Netherlands now from the 1st of January 2026, they will introduce a ruling on that. So that's quite a departure from a sort of more common approach. But I think people, across the board are seeing the need to have increasing control of the precautionary allergen labelling, because when people talk with consumers, that's the problem. The biggest problem they face is that inconsistency of what it really means in conveying the risk. And that's where organisations such as ILSI in Europe have proposed quantitative risk assessments as a framework for assessing that risk, and done some wonderful work and documents putting those guidance around that framework together. Definitely worth for people to look at those if they haven't already seen them.
And when you compare the European regulatory framework with the framework from other regions like in United States or Asia, how does it differ from these regions? Can we see there some clear differences? Or
yeah, I think for Adrian will also come in here, because I think we've both seen, if we look at, let's say, Japan, because I think Japan is a country which has taken an individual stance, and it's a very interesting position. And it started in 2002 with regulations regarding may contain labelling. They have a mandatory list and a recommended list. So there's 8 allergens on the mandatory list and about 20 on the recommended. And they've set a threshold at 10 milligrams per kilo of allergenic protein, and I think it's protein actually, I'll have to double check that. And each year they look at the list and the clinical cases which are being presented and seeing whether actually the list of allergens does represent what they're seeing in clinic. And they've added, over the years, they've added more allergens to the list. They've added shrimp and crab and walnut to that list, for example, over the years. So, but it's more than that, though, isn't it, Adrian? At the analytical level.
Well, yeah. But I'll just say about just going back about differences. Obviously, the amount of legislated allergens does vary from country to country and region to region. So in the US, they've just recently added sesame to the list. So it's now the big 9 instead of the big 8. Obviously, in Europe, we've got 14. In Australia and New Zealand, they've got different numbers as well, and South America have different things. I was just trying to think of the numbers off the top of my head. But if you there's a brilliant graph. If you go into the FARP website, there's a brilliant table that actually explains all the different regions around the world and all the different regulated allergens in that region. And it's a huge, great big matrix, because every country, every region has a different set of regulated allergens. Again, going back to it's specific for their local population, which is great for protect their local population, but makes trade a lot more difficult. And we see this a lot of the time in the UK, where you get imported products. Well, the product might have the correct allergen labelling from where it was made, but it doesn't have the correct allergen labelling for the country where it's being used, and we see that quite a lot. So so you've got this thing. We talk about harmonization. If obviously under Codex there's a recommended, a standard list of priority allergens. But then that it's up to the individual regions to change that. So it's good for their local their population, but does make this trade interesting. So I don't know how you sort of get around that. It's more diligence, I think, with the import-export. And I say, if you think about most of the recalls that happen are not because of what's in the food, it's because of the labelling errors. And historically, that's what most of the recalls for allergens has been, is because it was the the right product, wrong package.
Yeah, so harmonizing the approach is probably more helpful so that we can act regionally is probably more relevant to a country than a list, an ambitious list to cover the allergens for the whole world. I think that's what we're seeing.
So, Adrian, Richard, we are already coming to an end of this episode, and we are always looking a little bit also into the future in each episode. And Richard, you have mentioned already a couple of times the term artificial intelligence, and I would be interested now, which kind of emerging trends do you see in allergen detection? Do you anticipate some novel methods in the future, like DNA-based approaches?
I think, we deal with antibodies, and, these days there are alternatives to antibodies being looked at. So molecules which will also bind your target, aptamers, MIPs, so molecular imprinted polymers. So there are, whether these finally have useful application, we'll just have to wait and see. I think we could all, Adrian introduced earlier artificial intelligence. We've mentioned it a few times. And I think we all see the power that actually, when we look at small data sets, it doesn't give us the statistical confidence in results. And so when we have bigger data sets, more replicates, which as individuals, we always struggle with. But if we can pool data, and that is the biggest issue, is that ownership and that sharing of data. I mean, we know that regulatory authorities such as FDA and the Food Standards Organisation agency in the UK, they're already using AI as a predictive tool. But in order to use data, you've got to make sure that actually it is standardised, it is in the right format, a common format is so important. So yes, what are the developments? Cloud-based systems, bigger data sets, pushing up to the cloud, being accessed by more people to that data would give us more information and change, the power of analysis to be predictive. To be honest, I see that as the biggest step change, personally.
I think it's how that I agree. I think it's how that data is used and interpreted. As opposed, the methods will evolve, but if you look through the literature is littered with brilliant ideas that have never been commercialized. I mean, if you think of an idea now of how to detect an allergen, you can guarantee it was published about 10 years ago by a university, and it's just sat there gathering dust. So half the battle is how to implement those methods in the real world, I think. And at the end of the day, the issue you've got is we're dealing with a food industry that doesn't have a lot of money to spare to test. And obviously, when you go through a downturn, such as we've been going through, over the last few years, the first thing that gets cut is testing. So it's trying to be a bit more innovative, and this is something we've looked at. How can you do more with less? So maybe multiplexing more assays, multiplexing your immunoassay, your LFD or something like that. Put more lines on it. There are other complexities that come with that, because we talked about cross-reactivity. Well, if you put peanut and you put walnut and pecan on the same assay, well, you're going to get the one answer. So from my side, that's where the challenge lies, is trying to get pure detection without too much cross-reactivity to enable that to happen. But that's how I think we see the future going, trying to do a lot more with less money and less consumables, less plastic. Cut down. If you think about just look at COVID, look at all the LFDs produced during COVID, and where are they now? All these individual little plastic containers that are all littered the environment somewhere. So there's things to be done there, making it more sustainable, making the industry more sustainable as well.
Yeah, just to add, I think what we touched on there was targeted, which is what we deal with. We're going to detect peanut, and let's develop the method, and then also untargeted methods. And I think that again is another aspect to this. With the targeted, we could do more at the same time. So we could put more and more reagents onto the same spot, so that or we could do the untargeted approach. So I think there will be development in both of those areas. So that's really exciting, and the technology, which is supporting it as well. It's a technology-driven business, which is the thing I personally I think we get excited about. But we've got to remember, as Adrian said, it's the application and where it can be successfully implemented, and that's really key.
So to wrap up, what's one thing you wish everyone knew about allergens?
I'll go say, I said there's not one. I'll go for not one size fits all. To be honest, I think that is what we talked about, the toolbox. But there's no perfect method. There is no perfect method for detecting allergens, and that's sort of the it's always to question. I mean, the thing we haven't talked about is everything in the process of detecting allergens. A dialogue needs to happen. So it's no good just sending your sample to you're a manufacturer. You send it to the lab and expect to get a number back. The more you can tell a lab about the nature of the food that you're getting them to test, then the more information you'll get back from that result. So like I say, if you're sending them chocolate, then the lab knows, well, they might not get a recovery, and you'll know then you'll have to maybe multiply your number by 2, etc. So the more information you can give the lab doing your analysis, then the more confidence you'll have in that result. And the same is the lab. If the more dialogue the lab has with the method producer, the kit manufacturer, then the more confidence the lab will have in that result as well. So it's a win-win circular. It's win-win for everyone involved, as long as they talk to each other and share their knowledge about what they know about about allergens.
And for me, I think it is that lack of knowledge or desire for more understanding about sampling, which is my pet hobby horse, really. I wish I knew more about sampling, but because ultimately sampling determines what you find or you don't find. We've made tremendous advances with detection, but it's really, how to take the sample in the first place, whether it's a liquid, whether it's a solid, whether it's static, whether it's moving, this is where, there's statistics, which is a weak area of mine. So I'm not going to profess any capability here, but I think it's something we do need to do more work on, and top of my list would be sampling and how we can provide some greater guidance for people.
Yeah, thank you very much, Adrian and Richard, for sharing your valuable insights into this fascinating and also crucial world of allergen detection. I think, as we've heard today, while allergens remain a complex challenge, the combination of science, innovation, and especially collaboration, and maybe also communication, what you added now, Adrian, is steadily improving the detection, the prevention, and at the end also the public awareness. So I think for our listeners out there, it's also important to remember that the food on your table is safer today than ever before. And thanks to the efforts of industry leaders and experts like Adrian and Richard. And if you have enjoyed this episode, don't forget to subscribe to Analytically Delicious for more deep dives into the world of food safety and analytical science. Until next time, stay curious, stay safe, and keep enjoying the science behind your food. Adrian, Richard, thank you very much. It was really a great pleasure talking to you.
Thank you very much.
If you found today's conversation as enlightening as we did and want to stay informed about future episodes, remember to subscribe to our podcast on Spotify or YouTube. By subscribing, you'll never miss a bite of our delicious insights into food safety and analysis. And for those of you with burning questions or lingering curiosities related to our today's episode or any other food safety topic, don't hesitate to contact service at lva.at. Your questions will be addressed either directly in our next episode or by our team of experts on our website, www.lva.at. Thank you for tuning in, and until next time, stay safe and stay informed.